everybody, it's Haley, and today we're going to talk about genes. Okay, so first of all, something that you definitely need to memorize is that eukaryote chromosomes are made of DNA and proteins. So what are genes, alleles, and genomes then? Well, a gene is simply a unit of hereditary information consisting of a specific nucleotide sequence in DNA. Alleles are variations of the same gene that exist on the same exact spot in the chromosome. For example, different eye colors come from the fact that there are alleles that control that. So, if you have brown eyes, that's a variation of the eye color gene, and it's located on the same exact spot on the chromosome as like green and blue and brown eyes are all located, but they're just different. And lastly, a genome is the complete genetic information of an organism or virus. So that's all your DNA. So what are gene mutations then? Well, a gene mutation is an alteration to the DNA sequence that may or may not cause a change to the function of the gene, and you'll find out what that means on the next slide. So one of the IB questions is to explain the consequence of a base substitution mutation in relation to the processes of transcription and translation using the example of sickle cell anemia. Well, base pair substitutions, first of all, occur when a pair of nucleotides are switched with another pair of nucleotides, and there are three types of these substitutions. The first one is the silent substitution, which means that the nucleotide switch has no effect on the coding for the amino acid sequence. So like if one of the nucleotides gets switched out, it's okay because the new codon still codes for the same amino acid. The next one is the missense mutation, which means that the substitution alters the genetic code by transforming the nucleotides so that the codon for one amino acid gets changed into another. So if you had CCG and it got changed to CAG, then that would code for a different amino acid. And lastly is the nonsense mutation, which means that the substitution alters the genetic code so much that it transforms the codon into a codon that codes for a stop of the amino acid chain. So if it codes for a stop codon, then it just ends the amino acid chain where it is, and that's not really supposed to happen. So next is the sickle cell anemia part, and what happens is a nucleotide gets changed from adenine to thymine in the triplet coding of the sixth amino acid. And this mutation changes the allele HBA into a new allele HBS. And because of this single change, individuals with sickle cell anemia have valine in their hemoglobin instead of the normal glutamate. The name sickle cell, and that's supposed to say sickle cell, not sick cell, comes from the changed shape of the cell to a sickle shape because the mutation causes a phenotypic change as well as a genotypic change. And there's a good chance that this mutation would have already disappeared from the population, but in Africa, having sickle cell anemia actually helps fend off malaria. So it's very prevalent there. Quiz time. So first question, what are eukaryote chromosomes made out of? And you should have this one memorized. A, DNA and proteins. B, lysosomes and RNA. C, kinetochores and chitin. Or D, missense mutation and stop codons. A, obviously, DNA and proteins. The second question, which of these are variations of the same gene that exist on the same exact spot in the chromosome? A, genomes. B, chromosomes. C, genes. Or D, alleles. D, alleles. 3. Which of these is not a type of base pair substitution? A, a nonsense mutation. B, a genosense mutation. Or C, a missense mutation. Or D, a silent mutation. B, a genosense mutation, because all the other ones are obviously mutations. Thanks for watching!